Great pleasure and excitement, we go over to Lucy to hear about the DNA and the origins of Pictish bones. Thank you.
Thank you. Right. Oh, that's gone on a weird slide. Off to a good start. There we go. Yes, so thank you for that introduction. So as advertised, today I'm going to be talking about some new biological insights into Pictish identity. A disclaimer, I can't answer the question of who were the Picts in a big, grand way, but I can say a few of our results that we've been finding that give us a bit of an idea or a better idea than perhaps what Bede was saying about who the Picts might have been. So first things first, there'll be images of human remains in this presentation, just in case. So, and this is the structure of the talk that I'm going to be talking about. So, the project that I'm talking about mostly now is my PhD project, which the title of it was Investigating Migration, Kinship and Mobility in Prehistoric Scotland using Ancient DNA and Stable Isotopes. This started out as a nice grand idea, as all PhDs do, where I was going to talk about all sorts of different things. I was going to talk about the Neolithic, I was going to talk about the Bronze Age, and eventually that was far too much to do. So we settled on the Picts and proto-historic Scotland. And this was a collaboration both between the University of Aberdeen archaeological chemistry labs, but also the university collections, and also the Francis Crick Institute in the Ancient Genomics Lab at that institute in London, who we collaborated with for our DNA. And I was using isotopic and genetic analysis, focusing specifically on sites and individuals. So a lot of these studies that use archaeological science can be quite big, broad pictures that talk about, for example, the whole of Bronze Age Britain. My focus was on particularly on sites and individuals in this time period, and making sure that these individuals are not lost within broader narratives. And don't worry, I will explain all the science as we get to it, and I won't make it too complicated. So whilst I'm here talking to you today, this project was actually a collaboration between a whole bunch of people. So everyone I've pictured here is just the people who were directly involved in the project. There were more behind the scenes. So everything from Neil Curtis at University Collections, and then Professor Kate Britton and Orshi Sarah and Lena Skurdlam-Flink as scientists at the University of Aberdeen. But we also have scientists from the Francis Crick, including Marina Silva, who is really important in the DNA, Mark Oxon and Rebecca Crozier, and Jenna Dittmar as osteologists, and then, of course, the whole Northern Picts team, in particular Gordon, but also people like Edward, Nick Evans, and Juliet Mitchell who've been involved. So it was a big amount of teamwork involved in this project. So traditional narratives of Pictish identity, I've included here a picture of Bede, looking particularly like he's plotting things, have talked about various ideas about who the Picts might have been. In particular, you get this nice shift in the Victorian era where suddenly they're these enigmatic painted peoples. One of the big ones that has persisted and is always a question whenever I talk about my research is the idea of matrilineality that we've got from the Pictish king lists, where, of course, kings are named as sons of particular mothers, and various origin myths from Bede and Scythia to an Irish connection and the idea that perhaps the Picts stopped by Ireland to get their wives and then come to Scotland. And then also, of course, stuff about the shifting to Christianity. And a lot of archaeological evidence has since and specifically in the past, sort of 10, 15 years has come out much more investigating who the Picts were. So obviously this is the work of Northern Picts, who I'm sure in this audience everyone's aware of. I have this QR code here for people who aren't aware when I talk to other conferences and I have to convince them that Picts are cool, which is showing that there's quite interesting stuff going on as well. So we've got these massive high status fortified settlements where people are clearly gathering. So we've got Berghead here, and I think that's Tapanoth, and including also, as well as these massive settlements, we've got evidence for quote unquote exotic artifacts and nice things like bone pins and the Berghead ring, which show that there's quite a lot of elite production, but also trade and long distance connections. So why is biomolecular analysis, so DNA and isotopes, particularly useful for investigating these questions about who the Picts were? One of the big reasons is kind of the nature and the paucity of the historical and archaeological record. So historical sources are really slim. They're mostly written by people later than the Picts or outside of Pictland. And the archaeological record, whilst there's been massive steps towards this, we have these massive fortified sites, but the settlement evidence is still quite slim beyond these. And to be honest, even the cemetery evidence, there's a lot more, but even the skeletal record's not great because Scotland has acidic soils, which mess with everything. And in particular, a lot of these popular narratives are tying the idea of Pictland and Pictish culture to geography. They talk about mysterious origins, and they emphasize this influence of kinship in political structures, in particular this matrilineality. And we'll get back to the concept of matrilineality later because it's complicated. But there's also this evidence for gradual centralization of power with the overkingship, whilst retaining these regional power structures and diversity. So what biomolecular analysis can do is we go directly to the Picts themselves, skeletons dated to the Pictish period, and we can investigate directly questions of, for example, the origins of these people with ancestry, whether there's evidence for matrilineality, and all sorts of things like this. So these are some of the methods I'm going to be talking about today. So there's several key things. So osteology, which is the study of human remains, and particular seeing what we can get from human remains just by observing them macroscopically. And so things we can get from osteology, if we have a nice preserved skeleton, are things like biological sex or osteological sex, evidence of stress and trauma, health and disease, and how old that person might have been when they died. But if we want to get more information, we have to go destructive. So these are what we call destructive analysis, where we actually have to take a bit of the skeleton and destroy it to analyze it. And these are things like radiocarbon dating, which gets you when the individual died. And then isotopic analyses. So here we've got this is just the fancy way of writing carbon, nitrogen, and sulfur isotopes. And these we can use to look at things like diet and where someone might have been living in later life if we're looking at sulfur. Oxygen and strontium over here are more what we use to look at mobility. So this is residence, but this is in early life. And don't worry, again, I'll go all of this when we get to it. And then, of course, ADNA, which is sort of the big popular one, which is this deep genetic ancestry. So further back in time, and also things like genetic relatives and then chromosomal sex. So this is particularly important when skeletons are not also always not, it's not like it in things like Silent Witness where you look at a skeleton and you go, that's definitely male or definitely female. Often we get a skeleton, we're like, I'm not really sure. So chromosomal sex is the only way to know for sure. So there has been some previous research into genetics and isotopes in Pictland, although it has been quite limited. So I'm just going to quickly go over what we had so far. So the diet, and again, so this is carbon and nitrogen, which we can use to look at diet. The main conclusion from the limited individuals we have has been the idea that mostly people are eating terrestrial foods such as the classic sheep, goat, or pig, or cattle, even when we're near the coast. And also, so not much evidence for fishy Picts. And in particular, especially the work of Edward Massey-MacLean, cattle dominate fallen ore assemblages. So cattle seem to be really important in the fauna. And they're also, as I'm sure you guys are probably much more aware than I am, are featured on symbol stones, particularly at places like Burghead, although we don't tend to see domesticates like goat and sheep. So this is just a plot, and it basically tells you where all animals go. And so we see which was up here. In terms of mobility, this is also much less studied. So there's 3 case studies that I can find that have been previously published on this, from Kilfedder, Port Mahomet, and then work by Orshi Sarah on the Blair Atoll Man. Here you have my beautiful scientific graph. What I'm showing in this map is approximately what we've previously thought suggested mobility might have been during life. So people moving both east to west and potentially from outside Pictland into Pictland. So, for example, Blair Atoll and possibly coming from these 2 different directions into Blair Atoll, or people coming towards Port Mahomet and so on. And then an individual at Kilfedder who not only has moved to the Western Isles, but originally moved to, I think, Skye and then further on. So there's already some evidence that the Picts are not just tightly constrained to what we'd say is Pictland, and then moving around during childhood. And then this is the DNA. So this is the big paper that was published a couple of years ago. And this is based on London links, a few individuals from London links. And we've got mitochondrial DNA, which is you have more of, to look at things like haplogroups, which are an indicator of ancestry. And then we've got nice deep sequencing, which is what you need for these fancy graphs. For 2 individuals, this is a PCA. PCAs are really confusing, but essentially where you are in here is where you're most similar to, not just from like a carbon and nitrogen value, but from lots of different stuff. They've pulled out genetics, and that's shown that these 2 individuals from Ballantor and London links have ancestry most similar to modern Welsh and Irish populations, and similar to Orcadian genetics from the Iron Age and Viking Age, which suggests that these 2 individuals are not Scythian, as has been suggested, but consistent with the British Isles. And something to note is these data have been used in interesting ways. So these are some of the headlines we've had as a result of some of these analysis, where you've got really clear headlines. So some of them are pretty accurate, like Blair Atoll Man who enjoyed a diet rich in pork. But then this idea of modern day Pictish ancestry, and my favorite one is this one of Blair Atoll Man with his strong jawline. I'm going to tell you what my analysis suggests. Bear in mind that I've not got certain answers from this. This is just giving us a better idea. So I will not be able to turn around and definitely say this is exactly who the Picts are, but I can suggest things. So my PhD focused on 2 main sites from Pictland. Particularly isotopic work is ongoing from a lot of other sites across Pictland. That work's been being done by Cape Breton, and I know that some of that is in the works as well. So what I'm going to present from my 2 sites is consistent with this other new data as well. So the first one of these is Croft Gowan. So Croft Gowan is a monumental Pictish barrow cemetery, which is located at the base of Tor Alby, so up here, just south of Aviemore. You see here, along a nice curve in the hill, and it's on a major route between Stirling and Inverness, which would have been a major route at the time through the Great Glen. And Croft Gowan is particularly significant because it's believed to be, I think, currently the largest known Pictish barrow cemetery with over 40 barrows, which is enormous. And so this was excavated by the Northern Picts team, particularly Juliet Mitchell, James O'Driscoll, and Gordon Noble in 2021. And they excavated over a few barrows, but only 3 graves with evidence of human remains were actually found. And this is kind of typical of another problem with the Picts, which is these acidic soils. So we don't often get skeletons. So I will be referring to these individuals throughout as 410, which is this one here in Trench 4. And then in Trench 5, we have 518 and 515. So this round barrow is believed to be a later barrow than the others. And this is the kind of situation we were dealing with at Croftgowan. So we have these 3 individuals, but they were incredibly poorly preserved. So this is a really striking example of something called a body stain, which is indicating where that individual was. And you can actually see quite a clear outline of individual 410. So most of the bones were gone. We only have evidence from the skulls, from the heads of these individuals. This is 515. This is the best preserved individual. You can see evidence of the face here and the teeth and then the skull. It did not look this, it looked worse than this when it came out of the ground. So the majority of what was remaining was enamel caps, so literally just the crowns of teeth and some skull bones, which was difficult to work with, but we still managed to get quite a lot. And then the second site I'm going to be talking about is Lockhead Quarry. So this is just outside Gowanbank in Forfar. It's another Longkiss. This is a Longkiss cemetery, so not considered monumental, and traditionally not considered as high status, perhaps, although that's been contested, dating approximately to the 4th to the 7th century. And so, unlike unlike Croftgowan, this is not things, and this was also rather than being a planned excavation, as Croftgowan was a rescue excavation by AOC Archaeology Group in the early 2000s. And you can see that here. So whereas Croftgowan was nice in a field and relatively untouched apart from ploughing and so on, Lockhead Quarry was incredibly disturbed. As the name suggests, it's an active quarry. So first, in about, I think, April, what became Kist 20 was found, which is over here, believed to be a prehistoric kist burial. Prehistoric is in Iron Age, Bronze Age. And then a few months later, there started to be far more kists coming out of the quarry face. So where you can see these little crosses, this is where the kists are actually falling out. And there were a total of 20 long kists were found, although it's believed that there were more originally. And 18 had skeletal remains. And you can see here there's potentially some deliberate clustering at the site of graves. They all follow this very particular orientation. For 2 of the kists, it has the 2 that were empty, it's been suggested that they possibly contain non-adults, so children, based on the size, which would be considered extraordinary. I think we have to take this with a bit of caution because without the skeletal remains, we can't be sure, but there's the potential for that. And the key hypothesis of this has been that this was a family burial ground in some form, and potentially the clusters representing kind of family groupings. And so we have still poor macroscopic preservation, but much, much better than Croftgowan. As you can see here, this is the ribs protruding from the quarry face, and some of the teeth, but we have at least got a full jaw. And we have both males and females, although as seems to be a trend in Pictish cemeteries, there's a bias towards females, a bias towards young and middle adults. But again, so this is younger individuals up to the age of about 35 is considered, or 45 is considered osteologically young and middle adults. Although we have to be a little bit skeptical of that because the remains are in such bad shape. And One thing of particular note on the osteology of Lockhead is, whilst there couldn't be many conclusions, something that we have noticed across the individuals is something called linear enamel hyperplasia. So you can see this is these really distinct ridgy lines on this tooth, and you can see it from some of the others. And the reason it's significant is this is a sign of stress or illness in childhood when that tooth is forming. So for example, this canine, that would be between the ages of about 1 and 5. And so there's 2 discrete events, at least maybe 3. Traditionally, this is the easy interpretation of this is associated with lower status, lower social status. And there has been discussion of Lockhead Quarry, for example, being a lower status site because it is a long kissed cemetery with outstanding monumental structures. We will get back to that. So some of the questions I was asking in my PhD was, what is defining the communities we're seeing at these sites through various means? So looking at residents and mobility, whether the residence of these individuals in childhood is relevant to how much they belong to community in later life, i.e. are people who were non-local considered non-local in reality in death? And what distance from a site is considered local, but that's a complicated question we don't need to get into. Similarly, whether shared lifestyle or experience has been told by diet and stress and trauma can indicate belonging in a community and whether this reflects social status. And then also the ideas of whether biological relatedness is defining kin in these communities, and whether kinship is the most important social relationship defining inclusion in these cemeteries. So now on to the exciting bit, the results. So first, radiocarbon dating. So for Lockhead Quarry, I ran one more date. So this was already published in Dunbar and Maldonado in 2012 in the Tayside and Fife Archaeological Journal. So the period of use of Lockhead Quarry is about 370 to 780. But the main period of use is in the 5th to 7th centuries. Crofgowan, we got radiocarbon dates from only 2 individuals. 518 was far too, like it didn't work because there wasn't, the bones were not good enough quality. And we actually got 2 radiocarbon dates from 515, one from a tooth and one from a bone, which allowed us to do some fancy stats and make it shorter. And this is showing that these individuals were buried or died and were buried in about the 6th century mostly. So a very similar time period. Now on to diet. So the basic kind of background to isotopic analysis of diet is that you are what you eat, and that gets incorporated. So the foods we eat have particular values in carbon and nitrogen that get incorporated into our bones. And so here you can see this is how carbon and nitrogen is usually plotted, with carbon here and nitrogen here. And to make it simple, this is an example of roughly what we can see depending on where you are on this plot. So carbon, which is the one at the bottom, so how far you are along here. In the context of Pictland and early medieval period, we're mostly looking at whether the things people were eating were mostly terrestrial or marine, and possibly freshwater. So you can see that the terrestrial animals are all over here and about -22 to -18, but then the marine are much higher values, although they're all still negative. And then nitrogen, which is our y-axis, is also terrestrial versus marine, and also the trophic level of dietary protein. So how far up it is up the food chain, which is why we have these higher marine values, because before you get to the bigger fish and Pictish beasties or seals, there's a lot more little fish, basically. So this is a plot of the individuals from Crofgowan and Lockhead Quarry. So I've just averaged them here. So I've put little pictures of this, of where the animals would be. And these are all also carbon and nitrogen analyzed from faunal remains. So we have to do that to make sure we know what reference. And as you can see, all of these values are plotting up here. So this means and these are all adult values. So bone usually reflects the past sort of 10 to 20 years of life. So the diet kind of before the individuals at Croftgowan and Lockhead Quarry died was similar to fowl. So things like geese and chicken and ducks. And you can see here that there's you've got pigs here, and then because there's a trophic level effect in the nitrogen, there's usually a gap between what people are eating and where they plot. So this gap here, it's about 2 to 3 per mil, we call it, above pigs and sheep or goats, and that indicates that that's one trophic level, so that's what people are eating. So it's likely that the individuals buried in these cemeteries have a diet where they're mostly eating things like sheep or pig. Interestingly, they're plotting quite a lot higher than cattle. So even though we see a lot of cattle remains, it would appear that cattle is less important, because if you had a diet with more cow products in them, you'd expect someone to be around here maybe. And there's not much marine input. Now, one thing we were able to do for the Croftgowan individual was look at dentine. So this is your tooth root, and that forms when you're a child and it doesn't change. So this is the diet during childhood of this individual. So this is individual 515. And you can see it's a little bit higher in both nitrogen and carbon. And we'll get to that in the next slide. So this is the comparison of these individuals to other sites to see a bit more detail. So this is work that I've not put the names of the sites because a lot of this is ongoing work by Kate Relatedness in publication. And so here we can see that the carbon values are much more similar. So whilst both of these sites interestingly have like some of the lowest carbon, you can see that across contemporary sites, so across Pictish sites, there's actually really, really similar diet. So they're all eating despite, and like across Pictland from from Atlantic and Northern and Southern Pictland, diet is really, really similar, and there's not much difference. One thing we can note though, is that the dentines, the childhood diet of 515, is quite different and does plot outside this normal. And so this here, I'm pretty sure is Rosemarkie man. So this is potentially interesting because this could indicate more marine fish, could also indicate a slight difference of diet in childhood compared to adulthood. But this is one individual, so this is a clear point where we need to investigate further. But this is really cool. So it's suggesting that there's a lot of connectedness between the people in these cemeteries. And especially if you follow the interpretation that Pictish cemeteries actually reflect a limited part of Pictish society, the elite, this kind of tracks that there's this kind of shared dietary practices across Pictland. And yeah, this is just Lockhead Quarry with the individuals. There is still variation within Lockhead Quarry. So you have these two individuals here with not much high nitrogen, but slightly high nitrogen, which potentially indicates these two were a bit different, but overall it's very, very similar. And now mobility. So we use two different isotopes to look at this generally, strontium and oxygen. So strontium is actually named after the Scottish village of Strontium. And strontium, the values of strontium, which are just these numbers you see here, we get from the plants we eat, and it reflects mostly geology. And then the oxygen, because of the water we drink, H2O, oxygen mostly comes from the water we drink, so things like what local water sources we are. And the way we analyze these two is we look at teeth, we look at the enamel, which also reflects childhood. So we are comparing where someone might have been living when they were a child to where they were buried to see if they might have moved during their lifetime. And we do this by plotting out. So this figure here with all the colourful sections, each of these is an area with different strontium domains with lots of different strontium values. And this is mostly based on geology, but we also measure it from plants. So if someone had a value of say 7, 1, 2, they would be in these orangey areas. And that would be where they were consistent with living. And then the oxygen here, you see them, they look a little bit like weather pressure bars. And this is calculated from precipitation of the expected values you would see. So I'm going to do this side by side. So for Croft-Gowan, this is the data we have. So looking at this area around, these are the sort of values we might expect to get. And so what the carb means is this is the exact value we'd actually measure from our teeth, and then we convert it to drinking water values. And the key takeaway here is that pretty much all of these individuals fall within these ranges, which means that they are consistent with growing up locally. So it's likely that they didn't live too far away from the site when they were children. The one potential difference for this is individual 410. So individual 410, you can see here has a slightly higher oxygen value to the others, although it's quite similar. The strontium value is slightly outside this range, and also you see here the strontium concentration, and that's how much strontium we actually have in our teeth. Because the way strontium gets in our teeth, it basically piggybacks off calcium, which is what we're supposed to have because it's sort of similar, and then we end up with strontium. And this strontium concentration is much, much higher than the other two. So that can also tell us that actually maybe where they're getting their food is slightly different because they're getting this different amount of strontium. So we'll come to that in a second, but also what you can see here is for individual 515, we looked at all 3 molars. And so for individual 410 and 518 and all the other individuals I'm talking about, we were looking at second molars. And so this is representing where someone was living between the ages of approximately 4 to 7 years old. If we look at the first molar to the third molar, we're looking at everywhere from birth to up to about 12 years of age. And all these values are really, really similar. So not only can we say that this individual spent their childhood nearby, or likely did, but they also didn't really move around much in their childhood, which is nice to see because, as I mentioned earlier, there is some evidence for people moving in childhood. So this is a fancy plot where basically I use the values of 410 and we say, okay, it's unlikely that they're local. Where could they be from then? And this is I'm showing you guys the most exciting. So if I'm being optimistic, values, in reality, oxygen is a really difficult thing. So the oxygen, they could be closer by. But all these areas are places where this individual might have grown up. So you can see a lot of these areas are coastal, a lot of the west coast, but also up here in Shetland. And that high strontium concentration I mentioned. So high strontium concentrations have been associated in particular in Britain, especially over 200, with coastal areas. So it is possible that this individual from Croft-Gowan is growing up on the west coast or in island environments. That's nice. We're seeing at Croft-Gowan people potentially moving to the site and people who might have been growing up nearby. And then for Lockhead Quarry, so here what I've done is I've given you the ranges that we'd expect on the plot. So this is the strontium and this is the oxygen. So anyone who's within this kind of area or in between these two orangey boxes, they are consistent with residents in this area. So most of these individuals are falling within this box, and that box is for most of the area surrounding the site, the lowland areas and the valleys. So 8 of the individuals were consistent with that. And then another 2, which you see up here, have values which are more similar to these upland areas, so also fairly nearby. 4 of them did not have values that fall within there. So you can see that these 3 individuals, their oxygen is a bit higher. And then this individual down here, their strontium is different. So we're going to go to those again with the maps I showed you the same. So these 3, so KISS 2 and then KISS 9 and 17 are those individuals where the oxygen was different. And so these again showing places they could have been growing up. So KISS 2, we see again either these kind of islands or Western associations, or possibly even further south into the central belt and below. So this individual could have been moving from areas like Atlantic Pickland or further down into Northumbria and such. Picts 9 and 17, so they had very similar values. And again, they're showing these kind of Western potentially associations, but also further up into northeast Scotland. So there's potential mobility here within Pickland and then a bit further outside into Dariata and such. KISS 20 was a bit different. So as I mentioned, KISS 20 had different strontium values, and actually the values it had are very, very unusual in Scotland in general. So mostly down here in the central belt, a bit over here, and I think Mull. But if you look, this is in Scotland. If you add in Ireland, you see these values across Ireland. This does not mean this person did grow up in Ireland, but it's suggesting that this is one of the possible places where they would have moved from. And so it is likely, or a quite parsimonious explanation, that the individual in KISS 20 might have lived outside of Pickland in early life. And then DNA. So DNA is even more complicated and requires people who specialize in doing fancy statistics. But the general things we're looking at are chromosomal sex, so XX versus XY, and uniparental markers. Now, that's a fancy term for haplogroup, which is also a fancy term, but this is essentially a grouping that you're similar to, which suggests kind of your ancestry. So this is something you might be given if you did a DNA test yourself. I don't think Ancestry gives you this, but 23andMe does. So for example, my haplogroup is H1A, which is super consistent, super common across Europe. Marie Antoinette had it, completely makes sense. So with chromosomal sex, we can tell sex. And for uniparental markers, we can look at paternal and maternal relatedness. So you get your mitochondrial DNA down from your maternal line, and if you're a male, you get your Y chromosome DNA down your paternal line. And this is also an indicator of genetic relatedness and ancestry, because we see if people have wildly different haplogroups, then they're not so closely related, and so on. And particular haplogroups are common in different populations. So these are the DNA results. These were both a surprise and not a surprise. So there is mitochondrial diversity at Lochhead Quarry, and also with the individual from Croftgowan, we only got DNA from one individual. That is also a different haplogroup than we see elsewhere. So this is just an example of all the haplogroups. All those numbers just mean they're different. This shows mitochondrial diversity, i.e., there are not people from direct maternal lines in these populations, which potentially is an idea against a matrilocality, i.e. living close to where your mother is, which has been linked with matrilineality. There's also no close genetic relatedness. When I say close genetic relatedness, I mean relatedness up to the 6th degree. So for context, your sibling or your parent is a 1st degree relative. A 3rd degree relative is someone like a great uncle or grandparent. I could read you the list of 6th degree relatives, but it's really complicated. Great aunt, uncle, great grandparents. So none of the individual at Lochhead Quarry are closely related at all. It's unlikely there'd be an immediate family, which is really interesting. So this is not a family burial ground in the way that we might expect, which poses the question of what the hell's going on. I can't be sort of specific about this yet because it's unpublished, but 2 of the individuals at Lochhead Quarry also show differences in their ancestry to the rest of them. So most of the individuals show mostly British Iron Age ancestry, which is kind of what we saw with the previous paper. 2 of the individuals at Lochhead Quarry have something slightly different, and I found out about 2 hours ago that that paper, the massive paper that they're going to be involved with from the Francis Crick, is getting submitted in a week to Nature, and there'll be a preprint under this name out about then, which will have that information. So what is this telling us? And what does it mean? So we're seeing a connected and mobile elite. So even long-kissed cemeteries, which are considered non-monumental, a lot of effort's been going into them. They're in very conspicuous places in the landscape. You're having to drag massive stones. So most of the individuals we see in Pictish cemeteries are going to be elite, probably. And we see that they're connected. They have really similar dietary practices, which indicates shared cultural practices, because if you think about how we eat, things like feasting, sort of cultural food practices, this is very kind of related to that. And so this shows that the elite within Pictland, even if they might have been coming together over this overship, but sort of smaller pockets of power is very, very similar. And we also see that these elite might have been mobile, both within and beyond Pictland. So a lot of individuals are moving, but also a lot of them aren't. And it's a different scale. So, for example, some of the individuals at Lockhead might have been moving short distances, which is kind of what you'd expect, but not evidence of super long distance movement, again, from places like Scythia. And that we see that this diet and this burial is not really influenced by childhood residence. So none of the individuals stuck out if they were mobile. And it also speaks to the significance of women. So of the individuals at Lockhead Quarry, there is an even split between males and females in terms of mobility. There is also a 2-to-1 ratio of genetic females to males, which is showing that women are significant beyond traditional perspectives. So it's showing that women are also able to be moving around, but men are also moving too. But also that the women are being specifically selected to be placed in these cemeteries, which is kind of something we've been seeing previously, and Adrian Maldonado's done a lot of work on this. If these cemeteries aren't places where kind of kin in a traditional sense being buried, it raises the question of why people are being buried there. And especially Adrian has suggested quite a lot that this could be, as you've got this over kingship coming together, these places are a way of kind of enforcing community. So rather than representing a community that exists, it's a way of making one. And it seems like women are actually quite important in doing this. So coming back to the king list, actually potentially a female ancestor might be quite important in determining kind of power and stuff, even if it's not matrilineal in the traditional sense, which kind of confirms previous theories. But it's also showing, and the DNA is showing that whilst there's a lot of diversity in who the Picts are, ultimately they are likely the descendants of people who have been living within these regions already, rather than this massive new influx of people. So it's people who are already living in Scotland, with the potential for some other connections, and so on. So yes, I can go into much more detail if anyone has any questions, but that is my result.
Oh, thank you very much indeed. That was absolutely riveting. I mean, to get this sort of material actually factual rather than just imagined is so revealing, because I think some of the things that you were saying are kind of what we would expect, but you can actually prove it now, which is wonderful.
Exactly.
So are there questions from other people? As
Yes, we didn't, but now we do. So Lorna Carter is asking, could the diversity in ancestry be due to a religious community using the cemetery?
That is a difficult question. So this is something that's been explored with Lockhead particularly. In the original publication, it was actually to do with with early Christianity, because the site is very close to Restonov. I don't know if I'm pronouncing that right, Priory. The simple answer is probably not. There isn't evidence for a really strong religious community for kind of this early. It's obviously difficult because we have no settlement evidence. So this diversity is probably not linked to religious communities. I cannot confirm this, but yeah, it's probably not a specific religious community that's going on.
Thank you. While we're waiting for more questions, Lucy, are these cemeteries in special places in the landscape, and are they near to settlements, or are they apart from settlements?
So The first part of that question, yes, they are. And so Juliet Mitchell has done a lot of work on the position of these cemeteries kind of in the landscape, and they do tend to be. So they're not necessarily hugely kind of on the top of hills or anything like that, but they tend to be important places both for moving through the landscape and quite visible places. So Lockhead kind of doesn't appear to stand out, but it's actually quite a prominent position just outside Forfar. And if you were traveling through, it's quite likely you would have gone past it. Croftgowan similarly is super conspicuous. If you're going, like if you drive through the current road, you see it. So yes, and it's likely, I think, probably that's kind of significant, especially it's speculative whether they're kind of ancestry and IDing, but like they're making a statement, especially if it's quite selective who's going in. I've already forgotten the second part of the question.
Oh, it's like how close they are to settlements.
Yeah, settlements. We don't know because we don't have the settlements, unfortunately. This is the problem. So there's not a settlement right next to them. So it's not like where you might see in other places, like, for example, in kind of Anglo-Saxon places, we have settlements right nearby. But we haven't found them. So
no, that's always
potentially, but yeah.
Yeah, thank you. And then we've got a question from KJ Hutchison, who says, I might have missed it, but I saw the mitochondrial DNA results. Were the Y chromosomes tested, and could these people have been related along the male line instead?
Yeah, so Y chromosomes, it's difficult because that's only from the males. So there were fewer of them. They are very, very similar. They're all something like R1b, etc., etc. They're stupidly long sets of numbers. But that's really kind of common, basically from the Bronze Age to now, those Y chromosomes. So we can't really see that they're related on that in that sense. It's a little bit trickier to see, whereas mitochondrial, we've got a bit more of a lens that way. So it's possible they could have been related, but that's kind of trumped by the kinship analysis. So the Y chromosome could basically suggest that there's not much diversity in a population. So if you've got lots of different Y chromosomes, there's kind of more genetic diversity in that pool. But the fact that we've still got all of these people who are super not closely related suggests, then they're not really. That makes sense. It gets into complicated genetic statistics, but yeah, that it can also be used, but it's not as useful in that way.
So that makes perfect sense.
Can I ask something on that? Follow up on that. Is that sort of exogamy rather surprising if you think of Highland communities or particularly the fishing communities, even in the statistical accounts in the 18th and early 19th century, the ministers are sort of sucking their teeth and saying, oh dear, they're awfully inbred here, And there are many, many villages around Aberdeenshire where the minister is grumbling about the lack of marrying out. But maybe you've just got really quite small samples.
Yes, it's a combination. So we're talking about one site, and it's still we've got barely any Pictish genomes. So I think we're now from this we're up to, I think it will be 15, or we've got more coming. So that's still very small proportion of Picts. But it also kind of is linked to how these cemeteries are being used. So none of these individuals are closely related, and we also don't have a settlement they might have been from. And if they're being used by a specific subset of Pictish populations, like an elite, this group of people might be more likely to be mobile and moving around. So, and we see that in the kind of the idea of this traveling over kingship, and the movement of kind of goods. It sort of would track that there's actually quite a lot of mobility there. But also we don't know. So Lockhead Quarry, for example, could be used by multiple settlements across the landscape. We don't know because we don't have them. So it's a bit tricky to work out. Because if each of those individuals were of a different settlement, we don't actually know how kind of insular those settlements were.
You're really going to need more places and more
Yeah,
samples, aren't you?
absolutely. Yes.
Which of course will start coming when you're looking for them.
Yeah.
Brilliant. And Lucy, Tim Whittam asks, do you think that the diet at Croftgowan may have been related to the local geography, i.e. the marshland of the River Spey with high levels of wildfowl?
Could be. This is kind of where my expertise ends in sort of the local environment. It could well be, yeah. The interesting thing is it is in childhood. So the particular difference there. So that kind of relates to, it kind of suggests that maybe children are being fed something different, which we do see in other places. But yeah, wildfowl is one of the, so pig is the big one that's said as kind of a main protein source, but wildfowl is also highly suggested as a potential dietary source, both at Croftgowan, but in general. So yes, could well be that they're using kind of stuff nearby, including wildfowl.
Great stuff. And Duncan asks, are there more Pictish burial sites from which DNA/isotopic details can be extracted?
Yes. So isotopic details, there is a lot more forthcoming. So when I showed you that spread of contemporary sites, we have currently in progress in Aberdeen a massive dietary paper from lots and lots of sites. And similarly, we've been working on mobility from those as well. So that will be coming. It's just not guided specifically. So yep, DNA, as far as I'm aware, not much, although within that big genetic study, there are other individuals from across Scotland at the time. DNA tends to be a bit more tricky, but that doesn't mean that future research won't be happening.
Will there ever be a day when you can say there was a massive invasion of people from Dalriada taking over the Picts and removing their DNA from the whole gene pool?
I don't know, because the problem is we don't really have any people that we know of from Dalriada. So, we've got that's a really important thing to mention, is that this is the genetics of people who are being buried at this point in Pictland, but we already showed that they're moving. So there's no massive differences in ancestry based on mobility. But yeah, like these people are from different spots in Pictland, so we're getting a very broad picture, which is why the isotopic evidence is so useful, because if someone, for example, has massively different ancestry, but it turns out they're from southern England, that kind of makes sense. So yeah, we'd need a lot more isotopes. But again, that just shows you where people could have been moving from. It doesn't give you a nice geographic pin. And we'd also need some genetics from some proper Dalriadans, although that's,
who are they?
Yeah, exactly. Yeah.
Yeah, Lucy, Meg Massey asked, could you just please repeat the number of buried individuals you were studying?
Yeah. So Lockhead Quarry, it was 18 skeletons, but it was 17 for DNA, 14 for strontium and oxygen, and then all 18 for diet. Croftgowan, it was 3 individuals, but only DNA only from 1, mobility for all 3, and diet only from 2. It's a bit variable because what you can do depends on how much of the skeleton you've got. So to do mobility, you have to have teeth. You have to have a bone that has good enough preservation for diet, and then DNA is much more complicated. And if you've got decent teeth, it's great. If you've got the petrous in the skull or tiny little ear bones, but yeah, so it depends what you've got.
Brilliant. And then Neil Bell asks, if these are not religious communities, could the genetic diversity suggest a more meritocracy-based elite rather than a hereditary one?
Possibly, but again, it's tricky to tell. It's tricky because, again, we're not quite sure where the individuals from the cemetery are from and how they're being chosen. So whilst it could suggest that different people are elites, it could also suggest that people are specifically being chosen to be put into the cemeteries, who could be representatives, for example, of elite families. Which is, I can't really say which of those is more likely. basically. I mean, the other thing is that when we bury people, it's for the living, not the dead. And so people being buried in these cemeteries might not necessarily even be about who exactly they were in life and about their personal qualities, but potentially what they represent as people. So, for example, if they are members of specific families and so on, which is all speculative. But yes, basically, it's really tricky to say. So maybe it could be meritocracy, but we can't rule out because these sites could be representing quite a large catchment and quite selective inclusion. Because if we talk about even Pictish cemeteries without skeletal remains, there are far too few cemeteries to actually represent the Pictish population we think was existing at the time. So these are special sites with particular people being chosen to put in them. We don't actually know whether quote unquote normal Picts or whether whole populations were.
That's what I was going to ask, actually. Is like, do we know how the others were being, like, what were the kind of mortuary practices for the people that weren't selected for these sites? Do we know anything about that?
Not really. And, we make, so we call them elites. I would more say that these are sites where people are important in some way because people are putting a lot of effort into burying people in these sites. If Lockhead Quarry, all of these kits are lined by massive slabs of stone that are all the same. So that's a lot of effort. So the most likely explanation is elite, but we don't know where the other Picts are. And so that's also all of the stuff I've been saying about Pictish identity or who the Picts were. We've got to remember these are also quite specific people we're talking about. So it's quite possible that your average Pict isn't moving around, is eating far less protein. They could all be Scythians, who knows? But we don't know. Don't quote me, probably not. But yeah. So we can only work with what we've got. Yeah.
Yeah, sure. Brilliant. And Carly asks, what's the timeline range within a single site?
So Lockhead is about 2 centuries, about 250 years from the radiocarbon date. So it's possible that it's a much shorter period, but we can constrain it to those about 250 years. Croftgowan is super hard to say because we have there's more radiocarbon dates from things like charcoal at the site, but we literally have two radiocarbon dates from 2 skeletons from a site with 40 barrows. So that one, again, it's like looking at least 200 years, but we can't tell. But yeah, they're in use for quite a long period of time, likely.
Brilliant, thank you. And we haven't got any more questions. We've probably got time for one more if anybody has one. We've literally got one.
That's it.
Otherwise, we can wrap up.
Well, genetic. Are you getting one more question there?
Well, yeah, I think I've got a question from Meg. So, but it's over to little chat things. So, I understand that Denisovan sites have extracted genetic material from the soil. Is there the prospect for using the same techniques in Pictish burials?
Yes. So basically, you can analyze soil for genetic material, and the idea is you can actually look at soils in graves potentially and see the genetics of the person who's buried there. So, for example, at Croftgowan, where we have the body stains, we have actually got sedimentary DNA samples from those body stains. In Aberdeen, they collected them at the same time. As far as I know, they haven't been analysed yet, but I know Linus, my supervisor, was super keen on doing that at some point. So potentially, yes, there is the potential, particularly in that case that we've got, we're literally analysing soil that had this stain in it, so we're pretty confident it's that individual. Potentially, we could get some cool stuff from that. So that's something to watch in the future because that would be great. I think that'd be really cool. Yeah, especially from sites where we only have body stains, that could be really kind of useful.
Well, is that it, Fiona? Can we wrap up?
I think so. Yes, I think we can stop.
I'd love to say thank you very much indeed for a really pioneering piece of work. And I think what I found so exciting about it was that some of it is not yet fully conclusive, but you're absolutely at the sort of spearhead of technology. And what's going to come from now on. You're going to just get more and more data, and it will become more impressive and more instructive as work continues. So you're just getting the ball rolling right now, and here
Exactly,
we are
yeah.
listening to you saying this is the way it's going in the future, and that is really exciting. So good luck for your future research, and come back to Britain when you've finished in Flanders.
We'll see, yeah.